Okay, let's get started. My name is Dongeun Huh. I'm Associate Professor of Bioengineering at the University of Pennsylvania. And I'd like to start by thanking the organizers of the meeting for the opportunity to speak about our work today. Just as quick introduction, let me start by telling you a little bit about the work that we do in my research lab at Penn. So research in my group basically focuses on developing these microfabricated devices that we use to grow human cells, but we also use these systems to mimic complex structures and dynamic environment of human tissues and organs in ways that have not been possible using traditional cell culture systems. So here the idea is to use these systems as microengineered models of human tissues and organs for a variety of applications. So these systems are collectively known as human organs on a chip, and this term is used synonymously with tissue chips or microphysiological systems. So what I'd like to do today is to give you a quick introduction to this technology and perhaps help you better understand what this technology allows us to do using several examples of human organs on a chip model systems we're developing in my lab. So if you're new to this technology, perhaps one of the first questions you could ask is, how do we actually make organ chips? How do you go about designing and creating these advanced in vitro systems? So let me spend just a couple of minutes talking about the design principles that we use. So obviously you'd have to have a target organ, and for this discussion, let's say we're interested in modeling the lung. So the first step in this design process is to closely examine the anatomy of the target organ and identify the functional unit responsible for organ-specific physiological functions. So in the lung, that would be the alveolar system shown here. And the next step is to closely examine the functional unit to identify the key features that you want to replicate in your engineered system. So we often start this process by looking at the structure. So if you look at the alveolar capillary interface in the lung, there's a thin layer of alveolar epithelial tissue on one side, another thin layer of capillary endothelial cells on the other side. And these two layers are separated by a thin interstitium. And we also pay close attention to the environment of the functional unit. So what's unique about the alveolar system is that there's a blood flow through the capillary blood vessels surrounding the alveolar air sacs, and these air sacs expand and contract during respiration. So once you identify those key features, the next step is to design a microfluidic device that would allow you to mimic those essential features. So this is actually a human breathing lung on a chip system that we published many years ago. This is actually the photo of the device. If you look at the cutaway view of the device, there are two cell culture chambers separated by a thin porous membrane. So in this system, we culture human lung cells and capillary cells on either side of the membrane to mimic the structure of the alveolar capillary interface. We also use these hollow vacuum channels to stretch the tissue layers to mimic breathing motions in this man-made microfluidic device. So we can use these kinds of systems to mimic the structural organization of multiple tissue types in the functional unit of the target organ. But what's more interesting and exciting is that by having these multiple cell types and tissue types talk to each other, we can start thinking about mimicking more complex, integrated organ-level functions and responses. So to help you better understand what I'm talking about here, let's actually think about lung infection as an example. So during lung infection, let's say these bacteria actually make it all the way down to the alveolar air sacs. And one of the interesting things that they do there is to stimulate alveolar epithelial tissue to produce and secrete inflammatory cytokines shown as green spheres in this animation, and they actually diffuse into the surrounding blood vessel tissues, and they reach the blood vessels, and what they do is actually they activate endothelial cells in the surrounding capillaries. So when this happens, I use this movie to help engineers better understand this process. So these white cells, like neutrophils, circulating in the blood, as you know, they have the ability to recognize these surface receptors expressed by the activated endothelial cells. So what they do is they actually stick to the activated endothelial tissue, and they transmigrate through the endothelium into the alveolar air sacs. And once inside the air sacs, they start crawling directly towards the site of infection via chemotaxis. And when they get there, they try to resolve the infection by engulfing and phagocytosing the bacterial cells. So this is basically how our lung fights off infection. And as far as I know, this is one of the most complex organ level, not tissue level, organ level responses that happens in the lung. So the question we asked was, can we actually mimic this in this lung on a chip system? So what we did was we actually put fluorescently tagged the human neutrophils into the lower capillary channel, as you can see here. Nothing interesting happens, but when in the presence of bacterial cells on the lung surface, these neutrophils now stick, presumably because these endothelial cells are now activated. And this movie shows the entire process. So this neutrophil, as you can see here, sticks first, and then it crawls around. And at some point, it starts wiggling its way through the capillary cells and one of the pentagonal pores on the membrane to get across the three tissue layers from the lower compartment to the upper chamber. And once inside in the lung chamber, these white cells are shown in red here, chase the bacteria and phagocytose them, just like what happens in the living human lung. So we can not only mimic the entire process, but we can also visualize and analyze this very complex process in real time at high resolution. So this was the proof of concept demonstration of building complex organ chip systems published 10 years ago. So jumping in time, you might be wondering what's happening now. So one of the major trends in the field of organs on chips is to develop disease models and used these specialized models for a variety of biomedical applications, including drug testing. So just to give you a couple of examples of lung disease models we're building in my lab, as you can tell from the slide, we're actually developing lung on a chip system to study adverse health effects of cigarette smoking. So we have a very specific goal of investigating what happens in small areas deep in the lung during smoking associated or induced lung injuries and diseases. So for those who are new to this, this is what human small area tissue typically looks like. The tissue consists of the columnar epithelium supported by this stroma containing stromal cells like fibroblasts, interstitial macrophages, and also blood vessels. So to mimic this multi-layered architecture, we actually came up with this multi-layered microfluidic device consisting of 5 layers. So this is work done by Mark Mandrinas, who is now a professor at Tulane University. So to mimic this stromal layer, what we do is we fill this middle chamber with type 1 collagen gel mixed with human lung fibroblasts. And this layer is then sandwiched between these 2 chambers lined with primary human small area epithelial cells and lung microvascular endothelial cells. So using this system, we culture especially these area epithelial cells under air to induce differentiation. This is a micrograph of a fully differentiated epithelium that contains cilia, and also they form tight junctions. And we also came up with the way to embed these fibroblasts and culture them for long periods of time without the detachment or shrinkage of the gel. And as you can see here, these fibroblasts actually are sitting in the gel, and the starting material is collagen, and we're actually staining ECM, fibronectin, produced and secreted by these fibroblasts. So we have evidence that these cells start remodeling their surrounding matrices in this microfluidic device. So this is the setup that we use to expose the cells to cigarette smoke. We use off-the-shelf computer-controlled cigarette smoking machine that generates mainstream cigarette smoke that gets humidified and diluted more than 96% to mimic dilute concentrations deep in the lung, and the diluted sample is delivered to the smoking lung on a chip system. So the important question is, what can we study using the system? So there are a variety of endpoints we can measure and analyze, but in the interest of time, let me show you one interesting observation we made in this model. So just to refresh your memory, this is what the stromal tissue looks like with these fibroblasts sitting in the gel, and green color shows cell deposited fibronectin. So in one of the experiments, we actually smoked the cells with 1 cigarette a day for 1 week and 2 weeks and 1 month, and this is actually what we saw. We saw a very significant increase in the number of fibroblasts as well as ECM deposition. And in many cases, we saw these very dense cellular nodules and with a lot of ECM deposition around these cells. So this is actually, if you think about a typical fibrotic response. So this is nothing new. Airway fibrosis has been a common pathophysiological feature of diseased lungs in chronic smokers and lung cancer patients. But it was very interesting to be able to model this injury-induced fibrotic response in this organ on a chip system. In the interest of time, I'm going to have to skip over this, but we studied also adverse health effects of electronic cigarettes using this system. So here's another example of lung disease model we're developing in the lab. So in this case, we're modeling cancer immunotherapy for lung cancer. So with the support from Cancer Research Institute, we've been working over the past 3 years to engineer a system that would allow us to mimic immune cell cancer interactions in the context of cancer immunotherapy. So in this project, one of the first things we had to do was to engineer perfusible human blood vessels. And this is the device that we use to do that. And this is a cutaway view of the device. So what's interesting here is the fact that there are these two barriers protruding from the ceiling of the channel. So we use these barriers to trap liquid in the middle lane of this device. So liquid is injected into this middle lane. It nicely follows the lane. It never spills over to the side lanes because it gets pinned along these barriers due to surface tension. So this capillary pinning effect is demonstrated in this movie where black fluid coloring is injected into the central channel. There's a little bit of bulging, but it never spills over to the side lanes. So using this technique, we inject hydrogel solution mixed with fibroblasts and endothelial cells. And we culture them in the hydrogel. And during culture, endothelial cells in the hydrogel assemble themselves into 3D vessels that open to the side channels. So this vasculogenic process is visualized in this movie where if you look at this middle compartment filled with red fluorescent endothelial cells, over time you can see them develop a 3D vascular structure in the hydrogel scaffold. So this takes about 5 to 7 days, and at the end of this vasculogenic process, the endothelial cells form these beautiful tubular structures, and you can see these fibroblasts wrap around these tubes as if they were pericytes. And these vessels have open lumens, as you can see in this section. And we've also developed a technique to extract these vascularized tissue constructs as a whole after we form them. So these samples can be further processed for imaging. This is a scan electron micrograph of a single microvessel in the hydrogel scaffold. We can do TEM. So this is a TEM image of a single capillary, which is very similar to in vivo vessels. And we also have the ability to tune the architecture of these blood vessels. So depending on how many cells you put in, how stiff the gel is, these are all the same type of endothelial cells, but they show very different architecture during culture. And this micrograph shows anastomosis of endothelial lining in the side channels with the 3D vessels in the gel. So as a result of anastomosis, the vessels open to the side channels and the whole network becomes perfusible, as you can see in this movie. So this movie shows a fluorescent dye flowing through the vessels. And it's also possible to flow microbeads and nanoparticles for example, targeted drug delivery applications. We've also tested flowing blood cells, red blood cells on the right, on the left, and T cells. Actually, in this case, they are migrating in the TNF-treated vessels, and you can see all different sorts of behaviors, T-cell behaviors in this movie. So we're now using this as a platform to build more realistic 3D human tissues. So just to very quickly walk you through some of the examples, we've developed a vascularized human islet on a chip system shown here, where we put these cadaveric islets, actually we received from donors, into our device. We vascularize, and we can maintain their function for over a month actually without any loss of viability, functionality. We can also induce adipogenesis and vasculogenesis at the same time to model white adipose tissue. And this is a micrograph of a tumor construct that we got from our collaborator Scott Worden at CHOP, and we were able to vascularize these patient tumor explants in our device and maintain this tumor tissue for long periods of time. So what we do is we chop these tumor explants into small pieces. We put them into our device along with fibroblasts and endothelial cells, and over time they form these vascularized tumor constructs in the hydrogel scaffold. And as you can see in this confocal movie, the vessels are formed in and around the tumor constructs. And because the vessels are perfusible, the tumor constructs also become perfusible. And this is another platform where we actually create this open top. So in this system, the first thing we do is we put in this insert. We form the vessels following the same protocol. When tumor actually samples that are not easy to get become available, we bring these tumor tissues, open up the compartment, and put the tumor constructs into the chamber, into the hydrogel, and we vascularize them. So the nice thing about this design is that we get to determine where the tumor tissues end up in this device. And this is what the tumor tissue looks like right after we put them into our wells. And this is what they look like after a couple of days of culture. They become vascularized very quickly. And again, we can use these perfusible blood vessels in and around the tumors to flow cells, drugs, different kinds of materials for a variety of applications. So as I said, we're using this as a platform to study tumor immune cell interactions, and we have a lot of data, but in the interest of time, let me actually show you this interesting kind of proof of concept study that we conducted. So this micrograph shows mesothelin overexpressing A549 tumor spheroids, and we were actually able to vascularize these tumor spheroids in our device. So we're now using this model system to study the efficacy of engineered immune cells like CAR-Ts. So in this movie, these red cells are CAR-Ts engineered to recognize mesothelin expressed by the tumor spheroids. This is a couple of hours after infusion. As you can see, many of these tumors, CAR-Ts, adhere to the tumor-associated vessels, and within just a couple of days, many of these T cells infiltrate the tumor, and they're found within the tumor. And as a result of this T cell infiltration, we saw significant effect on the growth of tumors, and we also have evidence that T cells induce apoptosis of cancer cells to exert tumor-killing effects. So we're now using this system to screen the effects of different kinds of engineered T cells, and we're also delving into the molecular biology of tumor-cancer interactions. This is probably one of the most exciting but challenging projects we've worked on in the lab. So we've also sent those tissue chips, organ chips, up to space. We got selected as one of the 5 labs in the country about 3 years ago to send those tissue chips up to the International Space Station. So in this project, we're studying immunosuppression, which refers to this interesting phenomena that occurs in the microgravity environment. So it's known that for many years, that astronauts get sick more easily during or right after space flight, and there's some evidence that the microgravity environment compromises our immune function. So we're actually investigating this interesting space phenomenon using our model systems. So in this project, we're actually creating a model of respiratory infection, which accounts for more than 30% of all infections that occur in astronauts. And we're also developing a human bone marrow on a chip to model the entire process of immune cell recruitment from the marrow to the infected organ. So we've developed an array model, and we've also established array infection using a modeled microorganism, Pseudomonas. And we've also created the vascularized human bone marrow on a chip system using human CD34 positive hematopoietic stem cells. And this system really allows us to recapitulate the multicellular heterogeneity and complexity of the human marrow. So using these systems, actually, we've created this credit card-sized device, each containing 6 replicates of each organ. So 6 replicates of array on a chip, 6 replicates of bone marrow on a chip, and so this is the device actually we flew last year. So the system was sent up on SpaceX CRS-17 last year. And this is a photograph of the cube lab that contained our experiments up on the station. And we're in the process of analyzing data, actually using the samples that actually returned to Earth last year. So these are some of the examples of lung disease models we're developing in the lab. But if you think about it, this is not limited to modeling what happens in the respiratory system. So we can use exactly the same approach to mimicking any other organs in the human body on a chip. So to give you an example of such systems, we are also modeling the human eye, more specifically the front of the eye called the ocular surface. So if you're new to this, the ocular surface, as you probably know, consists of the cornea at the center surrounded by the conjunctiva, which refers to this transparent tissue covering the white part of the eye. Conjunctival tissue is also found underneath the eyelids. So if you look at the tissue structures, the corneal tissue consists of this stratified epithelium that has 7 to 8 layers. And this epithelium is supported by this collagen-rich stroma that also contains the cells called the keratocytes. These are normally quiescent stromal cells that live in the stromal tissue. And the conjunctival tissue contains these goblet cells that are responsible for producing mucus, which is one of the key components of tears. So another really interesting feature of the ocular surface is eye blinking shown in this animation. So as you can see here, blinking acts to spread tears to form this thin film on the ocular surface tissue that serves to hydrate the tissue layer, and it also forms this nice smooth refractive surface for light transmission. So this is something we found very interesting from an engineering standpoint. So to mimic these essential features, we've created this microengineered device that we call the human blinking eye on a chip. So this work was done in collaboration with 3 ophthalmologists at Penn, shown here. So this paper came out in Nature Medicine last year, so if you're interested, you can read more about it. And this is actually what the entire device looks like. We use this 3D scaffold, highly porous scaffold that looks like contacts. And so what we do is we, to mimic these multi-layered tissue architecture, one of the first things we do is to inject hydrogel mixed with primary human keratocytes into the cavities of the scaffold. So this micrograph shows nice deep penetration of the cells into the scaffold. And we've also came up with a way to deposit these 2 different cell types in a physiologically relevant manner, so we can actually put down primary human corneal cells at the center of the scaffold. And we can surround them with the conjunctival cells to form this concentric tissue pattern seen on the surface of the human eye. So during culture, what we do is we first seed the device, we keep everything immersed in culture media. We filled the upper chamber with the media. We flow media through the lower chamber as well. But once we form the tissue layer, confluent tissue layer on the surface of the scaffold, we aspirate liquid from the upper chamber to expose the cells to air, and we culture these cells at the air-liquid interface to induce differentiation. So during this culture period, the cells are fed basally from the media flowing in the lower chamber. So as a result of cell differentiation, as you can see here, the corneal cells that start off as a single layer form multiple layers, 6 to 7 layers, and so basically they become stratified. They also form tight cell-cell junctions, as you can see in this staining of occludin, which is one of the tight junction proteins. And the marker expressions. So cytokeratin 3 and 12 are terminal differentiation markers of corneal epithelial cells. So as you can tell, over time, marker expression increases, but it increases to a greater extent when the cells are exposed to air. And these are more interesting effects or observations. So initially the cell surfaces are flat, but as a result of differentiation, the corneal epithelial cells form these microstructures like microvilli, microplicae that are almost indistinguishable from those features seen on the surface of the human eye. We also confirmed that some of these conjunctival epithelial cells during ALI culture differentiate into mucin-producing cells in our device. So once the cells are differentiated, we use the system shown in this movie to mimic blinking. So this is gelatin hydrogel that we use as a mimic of the eyelid, and we use this electric motor to slide it back and forth to mimic blinking activities in human eyes. So this is a close-up of the movie. We inject contrived tears into this channel. We apply a little bit of pressure. The fluid comes out and gets picked up by this sliding hydrogel eyelid and then gets spread on the ocular surface. So the reason why we do this is to form thin film on the surface of the scaffold. We used OCT to actually image the tear film layer, which is about 6 microns, and this falls within the physiological range. Human physiological tear film thickness ranges from 5 all the way up to 10. So I'll just skip this, but we also did some modeling to show the similarities between human eyes and the eye on a chip in terms of the biomechanics. Initially, we were concerned about cells sloughing off due to blinking, but as it turns out, cells remain adherent and viable on the surface, and actually it helped the cells to differentiate much more rapidly. As you can see in this expression of cytokeratin 12, with and without blinking, we saw a more rapid increase in the CK expression in the presence of blinking in our model system. So based on this result, we speculate that the mechanical forces due to blinking might be contributing to cell differentiation and the maintenance of this tissue layer. So in this study, we also use this platform to model dry eye disease. And in the interest of time, I'm going to have to skip over this part today, but we basically use this system to mimic evaporative type of dry eye disease and use the disease model for testing ophthalmic lubricant. So I'm happy to tell you more about this offline if you're interested. And I'd like to just quickly mention that we've been also doing a lot of work in the area of reproductive biology and medicine. So, this is an area that has long suffered from a lack of human relevant predictive preclinical models. And so we're actually very excited about the possibility of contributing to this field using our organ-chip technology. So we published papers about placenta on a chip and a cervix on a chip again. I'm happy to tell you more about these model systems if you're interested. So before I finish, let me just spend a minute or two to talk about a new direction, a very exciting new direction we've been exploring over the past 3 years or so. So hopefully, I was able to convince you that organ-chip model systems are great in that They allow us to mimic the complexity of human physiological systems. But the flip side is actually, it's not very easy to work with these systems. It's a lot of work to develop these model systems. It's also a lot of work to maintain these tissue layers for long periods of time for experiments. So for these technical reasons, experimental throughput in typical organ chip model systems is very low. So I would say this is one of the most fundamental challenges we as organ chip engineers face in our field. So we took this problem to heart and began to work on developing higher throughput organ chip technology. And the way we do it is to scale up by walking away from those single channel containing devices. And we have begun to use these plate devices, each containing tens of replicates. And most recently, we developed this 6-inch wafer-sized device that contains 128 individually addressable cell culture chambers, which are regulated by very sophisticated pneumatic circuits. And this movie shows individual addressability of these culture chambers shown on the left portion of this movie. And we spent a lot of time on trying to automate the process of manufacturing these chips. This device contains more than 2,000 holes, and it takes about 4.5 hours to punch all these holes manually. So my student Andre, who's leading this effort, got frustrated, and he built this hole punching robot from scratch. So this is a robot convention. We use this rotating spindle to cut PDMS like butter, and between punches, the ejection pin comes out to pick up the core to prevent clogging. We can go through a large number of devices very quickly, as you can see in this movie. But we can also significantly enhance the quality of hole punching, as you can tell by comparison in this image. So we also automate fluid handling tasks in our model systems. This is a robotic loading of hydrogel cell mixture into the blood vessel forming device I spoke about earlier. So we can also automate some of the key steps of cell culture, like media exchange. So these systems, automated systems, are making our lives easier. But what's more important and exciting is that using these systems, we're now able to really drastically increase the amount of data, in vitro data, we can extract from these human cell-based model systems, which I think is unprecedented. We're very excited to further develop this technology in the next couple of years. Unfortunately, I don't have time to talk about organoid-on-a-chip technology, but in my lab, we're also developing microengineered platforms for the purposes of culturing and maintaining and also engineering various types of organoids. And again, I'm happy to tell you more about these model systems offline if you're interested. Just to summarize, this is what we do. We actually focus on developing microengineered models of human physiological systems, healthy and diseased human tissues and organs. So I did not have time to really go into this topic today, but in case you're interested in learning more about what this technology has to offer for drug discovery, I gave a TEDx talk 5 years ago about this topic, and we also wrote a review article for Nature Reviews Drug Discovery to talk specifically about the potential of organ chip model systems for drug development. So finally, this is my group, and these are some of the collaborators we've worked with, and these are funding sources. So I'd like to acknowledge their support on some of those studies I talked about today. Thank you very much for your attention.